Good afternoon, everyone. Thank you for attending this GenScript-sponsored webinar entitled Amplified Extracellular Vesicles Generation Triggered by Peptide Polymerization. I'm Dr. Minnie Hu, Product Commercialization Manager of Reagent Peptide Service at GenScript. It is my great pleasure to moderate today's webinar. In order to ensure that this webinar has high sound quality, please activate listening only mode in your GoToWebinar settings or mute your microphone. If you have a question for the speaker, please type it into the chat box for us to see. The speaker will be taking your questions at the end of the webinar. If we run out of time and he is unable to get to your question, we will be sure to email his response to you. So without further ado, let me introduce our speaker today, Professor Hiroshi Matsui. Dr. Hiroshi Matsui, currently a professor of chemistry at City University of New York, Hunter College, and also an adjunct professor at Weill Cornell Medical College, received his master's degree from Stanford University, PhD degree from Purdue University, and postdoc training from Columbia University. His work in the fields of nanotechnology and biotechnology and the integration of these two areas, for example, bio-nanotechnology, is aiming to produce creative sciences with high technology impacts, including engineering of novel drug discovery and delivery systems integrated from natural and synthetic nanoparticles, synthesis of extremely small iron oxide nanoparticles in various shapes and their applications, and mechanical study of biological systems. So thank you, Dr. Matsui, for joining us today. It's all yours.
Thank you. So, hi, everyone. My name is Hiroshi Matsui, and we're going to talk about the how to amplify the EVs triggered by the peptide and peptide polymerization, which is triggered by the enzyme cleaving and make the peptide to be polymerized in the cell and that carry over to the EVs. So quickly, so as Minnie introduced, I kind of joined a point with Hunter and Cornell, and this work is based upon collaboration also with the Kyoto Kashiba from the Japan Foundation for Cancer Research. My graduate student Mina Kan is also standby next to me. So in case you have any technical question, she can kind of help me to answer the question too. And as introduced, my background is engineering, more like engineer, and more physical chemist too. But now exploring to the medical and biological field. So my connection to the GenScript went long years ago, as my academic work started with the peptide self-assembly work. And that time I was trying to make some devices working with the self-assembled structure of the protein and peptide. But about 7 years ago, I decided to switch my research toward more medical fields, as my lab also moved to the Cornell Medical School. And ironically, I came back to the GenScript now. I thought that I graduated peptide, but for this type of the cell and exosome manipulation work, we actually came back to the peptide, and now we are again nicely working with GenScript, helping us a lot for the preparing peptide and understanding how it's gonna be affected biologically too. So. Well, she did a really nice introduction, but in addition, just the advertisement that we are also engineering the magnetic nanoparticle-based drug and gene delivery systems. And again, the characteristic nanoparticle shape seems to be affect the liver escape and also tumor site accumulation through the EPR. So if anybody is interested in this area, then we are always happy to talk about it too. So let us know. So the method presented today is the IP protected, but this presentation was more like the still progress report. And we are aware that there are other approaches to increase the EV production, like the hypoxia or pH or the sort of molecular injection method. And we haven't completed the proteomics in vitro and in vivo study for amplified exosome. So we still don't have a complete answer for that. And so we don't really identify the amplification mechanism yet. But so as many other the amplification method publication too. Although with the limited information we can gather from those papers, our method seems to be producing more exosome than other methods. So it's encouraging. And of course, again, these investigations are underway. However, our primary result seems to contain some novelty. And based upon this presentation and concept, we hope to further develop some collaboration with some of the listeners so that we can take further to this technology too. So the significance of the exosome, I don't think I have to explain too much to these listeners. So some exosome like coming from the mesenchymal stem cells, for example, they are widely applied to proposed applied to many organ injury, and also targeting the anti-inflammatory therapy. Also, some exosome derived from the dendritic cell or even T cell itself can actually activate the immune system, so that it's also proposed to be used as a cancer vaccine. So I think everybody is convinced that they have some high clinical potential and impact. But as I wrote in the bottom, the thing we need to overcome is the first large-scale production of exosome, or the EVs, and the storage and the collection. So those are the things that we need to work on. For example, exosome could, based upon the review article, the exosome could be harvested 1 microgram per milliliter in culture media. And typically the treatment require like a 10 to 100 microgram per mice, so that in clinical translation of this exosome as therapeutics, the amplification is essential. And we'll come to this point too, but what we're proposing could actually have also a bonus effect on the storage too, as you're going to see the cartoon later, but as the peptide is polymerized, and it will kind of make the artificial cytoskeleton in the cell and exosome, then it could actually enhance the stability, so that maybe we can store in the room temperature too. So that's another thing that we are interested in working on too. Anyway, so recently David Leiden's group, in my colleague in Cornell, and our group collaborates and report the heterogeneity of the exosome into at least 3 subgroups. We call it exomer and exos and exol. And these subgroups have unique functions and biodistribution and packaging of the proteins and nucleic acid like DNA, RNAs, lipid, glycans, and but also we are interested in the mechanical property diversity of these exosome family. And as I wrote in this slide, the size are different. So exomer is very small, exol to be larger, and exos is more like a canonical exosome, with the quite diverse stiffness between them. So our initial motivation was that we want to tweak the stiffness of these exosomes, so that that could actually change their functions. And that's why we come to the idea that maybe peptide and polymerized peptide triggered by the enzyme could be useful for this purpose too. By the way, before going to from this slide, I'd like to just define the terminology. So only for the presentation and from now when I talk about the exosome, which means that this is the EB, we separate it from the 100,000 G. And when I mentioned the EB from after this slide, meaning that this is the larger particle, we separate with the 12,000 G. So that's the sort of terminology I'm going to use it from this slide. Okay. So in this work, the peptide we're using is this sequence, it's shown in this upper portion of the slide, and this peptide is synthesized by GenScript, and it's conjugated with the MBD dye, and this structure is originally designed by the Bin Shu in the Brandeis University. And we have also close communication with them, and we kind of got some help from them too. But so we're using this peptide, and so this peptide, as shown in the green circle here, once enter the cell, the entrance of this precursor to the cell is we don't treat in any specific way. It seems like it's incubate in the culture media and naturally go inside. So we don't do a special treatment for the introduction of this peptide. So once this peptide precursor goes in, then alkene phosphatase, the LP, will cleave in this phosphate group, which is making this peptide to be more hydrophobic and start polymerizing. So as I wrote in this sort of green spike, it starts polymerizing and forming like artificial cytoskeleton. And since we have this MBD dye, which is designed to emit the light when they polymerize it, so we know the color change so that we can notice that they are polymerized. Then we secrete the exosome will be corrected. And then we'll find out how these exosomes are different from the original one. So again here is how in the fluorescence micrograph look like. So once again peptide cleave and polymerize it, then you see the color change. And in this case, the color change to green, and sometimes you could also see that the peptide aggregation itself, without really in order the polymerized structure, they could light up to the red color too. And sometimes just for your information, sometimes these color could be flipped, and it depends upon the cell type and the cell density, and also the pH, and many things will actually change the color. So at this point, the most important thing is that you see the color change, and that's more important. So here we stain the actin, and the overlap of the actin and the polymer is shown as this yellow in this fluorescence micrograph, and the nucleus is as blue. So as you can see that the overlap is quite significant, and it seems like this polymer is growing around the actin, and so that's why you see this overlap going too. So the way we actually evaluate the stiffness or mechanical property of the exosome is using the atomic force microscope, and using the so-called nano indentation. So basically cantilever pushing the exosome, and we measure the force and how much we need to push to deform this exosome so that we can get these sort of the force curve. And this is an example that we look at exosome for the melan A versus the more aggressive the melanoma exosome. And roughly speaking, the slope will be proportional to the stiffness. So if you hit the silicon substrate, it's going to be extremely stiff. So you can see the slope goes up. And then so these two exosomes is obviously have the different stiffness that you can see. So based upon that, for example, we look at the F10 B16 exosome and do the AFM indentation. And this is still primary, but if you compare the exosome without polymer versus the treated exosome, the stiffness seems to be increased. So indicate that the exosome will carry over some amount of the peptide polymer from the parental cell. But when you look at the size distribution of these exosomes derived from the MB231, I'm sorry, the F10, you see that the distribution is very similar, so that it seems like a size doesn't have a major change with and without peptide polymerization to the parental cell. But again, the interesting is happening, and what we found is that it's not just the stiffness, but the number of the exosome coming out from this parental cell treated by the peptide have this significant difference. So in this graph, the x-axis is the concentration of those peptides incubated to the MB231 in this case, and we count the number of the exosomes. And it turns out that as the concentration of peptide increase, then the number of exosomes coming out from cell increase. So we thought this is interesting. Maybe we could use as amplification of peptide if the packaging is not that significantly different. And so this is a kind of quick screening of the exosome generation by the BCA analysis. And we try right now the 4 different cell lines, and M231 and 1080 show the significant increase of the exosome production based upon the 100 micromole of the peptide injection to the parental cell. And interestingly, the F10B16 and MB231-4175, which is lung-tropic breast cancer cell line, doesn't have too much difference, or the MB231-4175 case that there's actually no effect, and which does make sense because those cells doesn't contain high amount of alkene phosphatase. So this is kind of the control experiment that in this peptide we need the alkene phosphatase, which how we designed. So it's supposed to be that way, but this is showing that it's really this phenomena is occurring based upon the enzyme cleavage of the peptide and polymerization. But if you look at the fluorescence spectra of this solution of the exosome, then when you see the increase of the peptide concentration, the fluorescence intensity increase too. So since number is also increasing too, and size has not much major impact by the peptide incubation, so this will indicate that the exosome is coming out from the parental cell also carry some peptide polymer fragment inside of the cell or the outside the cell. So here is the TURF micrograph. And these are high resolution fluorescence micrograph of the exosome, which is treated, the parental cell is treated by the peptide. And as you can see that in TURF, you can see that those spot, which is showing that the exosome is really containing some kind of fluorescence material. And in this case, it has to be this peptide polymer. And also the MB231, this is after we separated to the exosome, we still see in the MB231, HT1080, you see this spot, which is fluorescence, which means that these seems to contain the exosome too. And well, still again, Steve, this is a work in progress, but we quickly do a Western blot to see that how much the packages were affected. And so far, we did the M231, H21080, and the most of the exosome marker, which exists in the exosome without treating the peptide, will also contain in the peptide-treated exosome. So all showing the yes and yes and so on too. Interesting thing is for the MB231 case, the control exosome doesn't have too much the actin, but the actin level seems to be increased after peptide treated the parental cell of MB231. So we need more study, and we need to diverge more cell types, but so far it looks like packaging, it seems to be very similar. So for these, for example, for MB231 and H1080, we saw that actually ALP, the phosphatase, is also contained to the secreted exosome. So we really want to, let's say somebody want to change the stiffness of the exosome, but without changing packaging, but not worrying about the amplifying the number, then maybe what you can do is that you can directly incubate these peptide into the exosome, and since there's still enzyme in it, so in theory you can produce these peptide or sort of artificial cytoskeleton grown exosome in the exosome too. So what's next? So again we are looking at these exosome treated by the peptide, but as I introduced in the beginning, the exosome is already, everybody knows that it's heterogeneous, and just the normal exosome themselves are heterogeneous too, like exomere, exos and exo-L, probably more than that. But then if we treat by the exosome, then they should also trigger the heterogeneity too. So we are interested to look into more those peptide-treated exosome in heterogeneity in terms of the packaging and population and size and stability, and we'll find out how those are changing. And I just want to point it out that further on this heterogeneity study, we may find that some exosome subpopulation treated by the peptide could be, the amplify in significant number, but maybe the peptide, the protein packaging may be identical, or maybe some portion, maybe they can be grown, and they can be similar packaging, but they may not contain the polymer, which maybe some people want more because this could be more applicable to the patient. So we are hoping, and actually we have some indication that that could be the case. So we are hoping that maybe we can find a subpopulation which is again the amplify, but that could be having a package, or that could be the property and the function that we want, and we can isolate them too. So this is just kind of future direction. And in a more clinical aspect, that that could be polymerized by the other enzymes. So it depends upon the cell type. Maybe we can tune it. But we also start seeing whether the peptide can be polymerized, but without these attachment of the dye, because at the end, once we know the mechanism and we understand that what type of the condition, we can always polymerize it and then drive the exosome, then we probably don't need the dye anymore. So we'll hope that we can develop this venue too. And lastly, we have some primary observation that the number of EB is also increasing with the peptide incubation, and the packaging is slightly different. So we are also interested in to see what's happening to the larger EB side of the effect under the influence of incubation of the peptide polymerization. So, okay, so this is last. I think that's give it to Minnie to explain this, right?
Thank you so much, Dr. Matsui, for such a wonderful presentation. So we will now move on to the Q&A session of this presentation. To the audience, please take a few moments now to type your questions into the question box. While you're typing in your questions, I would like to introduce GenScript's peptide synthesis service. GenScript has a comprehensive peptide synthesis service, including custom peptide synthesis with regular and express synthesis that can deliver peptides in as fast as 5 days. We also provide peptide library synthesis service, including standard and microscale peptide libraries, as well as large-scale peptide synthesis service, including cGMP and cosmetic peptide synthesis. With our PEP-power technology, we are capable to exceed 98% success rate and synthesize sequence length up to 200 amino acids, which can be delivered with 100% guaranteed quantity under total quality management. Over more than 15 years, we have delivered over 600,000 peptides to more than 10,000 scientists worldwide and helped them accomplish over 3,000 publications. GenScript's team of peptide synthesis experts are always looking to help researchers achieve their research goals. Please review our comprehensive set of reagent services on our website, www.genscript.com. All right, with that being said, we will start our Q&A session. So we have question coming in. The first question is, how general is this amplification?
Yes. So again, so far we are seeing the 4 different cell types. We are looking at more, but again, in this case of peptide, we need the alkaline phosphatase. So as far as the cell has the alkaline phosphatase, then in principle that should be applicable. So it should be general based upon this, the enzyme inclusion. And we are still testing more and prove our concept. But so far we did a 2 cell type and working on another 2 cell types, and it seems to be consistent.
I see. So the second question is, does this amplification happen for both EVs and exosomes?
Yeah, so this is the preliminary result, but so in the beginning we saw the entire population was increasing, so we were wondering what's going on, and then we start looking at in different size and the ultracentrifuge to separate, and it turns out that those proportion seems to be increased. Yeah, so both are increasing. So I guess that's the answer. Yeah, I was gonna go into more detail about how much they increase, but maybe still this is under the progress. But the bottom line is both are increasing.
Great. So we have a third question. How are the packaging difference in EVs and exosomes before and after peptide amplification?
So so far, as of now, what we're seeing is that the packaging of the exosome part is very similar with and without peptide polymerization, but EV seems to have the more peptide polymer. So in other words, EB is brighter than the exosome, and we have less proteins in the EVs for some reason. So in that sense, it's interesting. It may indicate some biogenesis of it, but it's a quite contrast compared to the exosome. So yeah, EV is interesting in that sense.
Great. Another question: are there any other peptides that can be used for the EV amplification?
Yes. So again, there's even the community of these peptide-based hydrogelation, and again, the Binshu was the one of the well-known and active person working on this. But yes, there's a peptide design that they have a bond cleaved by for example, phosphatase or protease or esterase. And so once you design this peptide and cleave and maybe polymerization, then basically that can be used. So the point is that there's a multiple peptides available, and each peptide has the different function to be cleaved by different enzymes. So if you have a library, then probably we can design a peptide which is specifically polymerized by the specific enzyme.
Great. We have a lot more questions coming in. So the next one is, have you tried to increase the production of EVs in a bacteria model?
No, we haven't. Oh, that's an interesting question. Yes, we haven't done it. Although we did actually amplification of the magnetic bacteria for a while ago, a couple years ago. Although we haven't completed yet, but frankly, I don't recall that there was an exosome the number of exosomes increasing or not. But in that system, actually, the existence of exosome is not even discovered yet. So we found that we identified that those particles are not really the fragment; it's just really exosome, and the magnetic bacteria's exosome actually contain the magnet. In this case, the iron oxide. So that was the previous discovery. But don't recall that we count the number of the exosomes. So, but they produce the exosome for sure. Yeah.
I see. So what is the length of the synthetic peptides? And also, can we expect an immune response against the peptides if delivered in vivo?
That's actually those toxicity study and those in vivo study is done by the Bin Xu who designed this peptide and publishing. First publication coming in Nature Communication and it's coming through. So yeah, you can take a look at his paper, and he did more well, he doesn't do exosome, but in terms of peptide toxicity, then he done a lot of those based upon this peptide. So you can take a look onto his papers.
Great. And what is the importance of exosomes or EVs with more stiffness at a clinical application?
So we only have a hypothesis, and nobody knows yet. But at this point, only I can say is that the stiffness. Oh, because I showed that the 3 subgroup of exosomes, and they are different. They have different stiffness, and they have a different function. So there's a possibility that the stiffness will affecting those function. So if we change the stiffness, then function could be changed. And this shouldn't surprise to people who study all the stiffness of the extracellular matrix these days, because it's getting very hot topics that the stiffness of extracellular matrix is very important, and that could develop the tumor or the cancer microorganisms too. So I think based upon these tissue and extracellular matrix stiffness, there shouldn't be no surprise that the stiffness of exosome might have some effect on biological function too.
And how important is ALP for exosome formation?
Well, in this design peptide, the ALP is essential, because if ALP doesn't cleave the phosphate bond from the peptide, then they don't polymerize it. So that's the enzyme trigger the polymerization. So all of this system actually need some kind of enzyme, but we can actually design the peptide sequence to be specific to certain enzymes. So let's say you're interested in the tumor, and obviously many people now talk about MMP9 enzyme. So it can be designed to cleave the protease with, so that these peptides can be polymerized in those specific enzymes too.
And next question is, has this method of using peptide-based amplification on increasing the yield from primary MSCs?
No, we haven't done that yet, so you can't answer that question.
Okay. Next one is in peptide amplification. Can cancer cells be treated?
Meaning cancer cell can be targeted?
This can be treated.
Treated. So the cancer cell can be treated by specific exosome, for example, like a T cell driven exosome. Is that what that means? Then the answer is yes. But so depend upon the what exosome, right? So if again, as I show in the introduction slide, if you're interested in the cancer vaccine and maybe exosome carry the sort of the same function, then the exosomes can be used to treat the tumor, for example. And some, well, but those exosomes need to know where to target too. So, and that could come from different functions. So I guess the if you want to treat certain disease with exosome, then you probably need a combination function. Some are targeting and some are curing. Right? So somehow you have to engineer that sort of those multiple function in the exosome, or you can maybe generate in natural. I don't know, but we are thinking that we have to engineer.
Great. The next question is, so how exactly can you be sure that a given extracellular vesicle is an exosome and not something else?
So the I guess something else, meaning like a protein fragment or the sort of junk, I guess. So that we can see one way to do in our lab is that we always look at the stiffness, and stiffness is completely different. If you look at those sort of the protein aggregation or some kind of junk aggregation, they don't have even stiffness. So the we can tell the population wide. We can't, well, because we can test every single particle and separate it, but we can kind of see that how much it's contaminated or not.
I see. Is there a dose-dependent response of peptide treatment in regards of beta-actin?
It seems so. So we only do the 2-cell line, but the so far it looked like that way. But we only try. Let's see. We only try 3 different concentrations for that. So the again, the data point we need more, but the as of now, in small number of the data point, then the answer is yes.
Okay, so we have a lot more questions coming in. So let's take the last 2 questions. Can the peptide polymerization alter the exosome capacity for drug delivery?
Capacity. So if you're talking about the biomarker-wise, then so far what we're seeing is that the packaging is similar. So that the best way to look at it is that the key proteins, which is known to adhere and target certain organ or tissue, and make sure that those proteins are expressed, and if they do, then the things should target it. Like for example, if you target the lung, then the integrin is essential, and so if these key integrin is expressed in these exosomes, and you can confirm it obviously, then it should be still function, and yeah.
Great. And what would you say are the most important issues in exosome research?
Repeat again, please.
What would you say are the most important issues in exosome research?
Well, again, as a sort of from the engineering side aspect, as I wrote in the introduction slide, I think the well, based upon the certain exosomes is known to function and target, and or maybe therapeutic effect is working, then I think that, again, the increasing the number by amplifying is important. And again, the storage is important too. I think the you can have it. Well, that's happened to our lab too. We always wanted to use exosome in a fresh condition and make sure it all function, because it could be degraded. And so if the storage is easier and it lasts longer, then that certainly help to those therapeutic exosomes to the clinical translation.
Thank you, and thank you to all for all the questions. And unfortunately, we are out of time and have to conclude this webinar. Once again, responses to any questions that we are not able to answer right now will be emailed to you directly. Thank you very much again, Dr. Masuri, for sharing your research with us. And thank you to all who have taken the time to join us. See you next time.